to point out that tomorrow is the occasion of George's commencement, or whatever the equivalent is called at the moment. It's not proper commencement, so he and I will be at the Coliseum celebrating that. So congratulations again on that. And it reminded me that I met you, George, I think in 520, right? I think in stats computing. And I labored under the belief that I had anything to teach you about computational for a week or two. And you rapidly disabused me of that notion and have continued to do so. So to the point where you're now applying for a job in the department, which is super cool. So this is by way of a job talk, in case people don't realize. And the subject is automatic annotation of gene function and GEESE, and it permits me to make a dad joke, if you don't mind. I'm very confident the talk will go well, but if it didn't happen to go well, it means we could say that you've cooked your own goose, right. Which is an expression you may have to look up, but I was trying to check the etymology of, and there's no known origin for that expression that's believed, but I couldn't resist the joke. Anyway, George, it's been a pleasure. So over to you, and let's hear what you've got to say.
Thank you, Paul. Well, it's great to be here again. I've been giving talks to you guys a long time now, so this is not new for me, I guess. But the difference here is that today I think I'm going to give it a bit more emphasis to the details. I usually, when I give talks about my research, has been in the context of like talking with other researchers and presenting others' work. So I try to profile down a little bit my things, my stuff. But now I'll give enough detail that I hope you will enjoy it. So let me start sharing the screen. And I apologize in advance because of the fact that for some reason they haven't fixed Zoom for Unix, for Linux yet. So every time that I share my screen, people tend to see like a black box, which is the typical black boxes that are invisible for the rest of the Zoom users, but not for me. So I don't know why, but so stop me if you see have any question, or if you can't see anything properly. Let me see. Okay, can you see the screen?
Yeah, that looks good.
Okay, so today I'm going to be talking about gene functional evolution using SufficiEncy, or GEESE, as I like to call it. I've been kind of, I've noticed that a lot of people in bioinformatics tend to have cool names, so I didn't want it to be left out, and I tried my best to come up with a nice name for this, and I think this is a nice name, but we'll see in the future if people stick to it. So for today, I'm going to split the presentation into 3 parts. The first part, I'm going to be describing a model that we already published in PLOS Computational Biology, which was a key part of my dissertation, an evolutionary model that it's very simple, but yet super scalable. You'll see some results on that. Next, I'm going to be talking about a new iteration of that, which is actually a generalization that is based on using SufficiEncy in order to model more complex features of the evolutionary process of gene functions. And finally, I will end up showing you some preliminary results in which I compare these 2 models, aphydo versus GEESE, using real data. Okay, this is joint work with Paul Thomas, Paul Martram, me, Duncan Thomas, and John Morrison. So the main question here is that what we want to know is, we usually are interested about whether gene XYZ, for example, is involved in some process, call it ABC. The thing is that in order for us to do that, everybody, in this department is trying to go after what particular genes do, or which genes are involved in what process. And as you know, it's very complex to actually try to assess that directly using in vivo or in vitro experiments. But at the same time, we actually do know quite a bit of some other species regarding what their genes do. So for example, the fruit fly, which is a very widely used biological model, we know a lot about it, and it's being widely studied. And we know about whether, for example, some sets of functions, they might have some functions, which functions they don't have, so on and so forth. And at the same time, we actually, while we don't know much, quote unquote, about the human genes, what they do, we actually have a pretty good idea about how genes are connected throughout evolution. So as you know, at any time that anyone thinks about the tree of life, you think about I don't know, we're connected with the monkeys and things like that. But in modern times, thanks to gene sequencing, we actually are now able to connect species throughout evolution based on their genes. So instead of having, I don't know, how humans are connected with, I don't know, monkeys, we do know how each particular gene of a human is connected with each particular gene of a monkey or a mouse or a fruit fly, so on and so forth, at that detail. So the thing is that we can actually use this to help us answer the question about what genes are involved in process X, Y, Z, ABC. So let me, to rephrase the question, what we are looking for is to answer, is the human gene XYZ involved in process ABC, given what we know about that for other related species? In other words, like a conditional probability. And this is the kind of like the punchline of this, leveraging our knowledge about other species and the way that they're connected with humans to make predictions or assert the presence of absence of function in other species. Regarding the data that we use for this, we have essentially there are like two big sources of data. The first one is the gene ontology, of course, in which we have which is connected to the Panther project, which is directed by Professor Paul Thomas and Professor Wayu, in which we have of these trees that you observe here, in which we connect species throughout evolution and their genes throughout evolution. We have roughly 1,500 phylogenetic trees, which are like organized in terms of families. Besides that, from the gene ontology, we do have about 8 million annotations of different genes. And by what I mean with annotations, I mean essentially, I don't know, this gene is involved in process XYZ. This gene regulates this other process. So we have that sort of annotations. Of all of those 8 million annotations, 600,000 are on human genes. Yet less than 10% of the annotations that we have available are based on experimental evidence, which makes it very complicated. And that's because of what I mentioned earlier, right? So trying to assess this directly in vivo or in vitro is very complicated. So we tend not to do that. And because of that, at the same time, we are actually in a race to improve our biological knowledge, to advance biomedical research. And what I mean with that is that essentially all these annotations that we have been generated by automatic methods because we are in a hurry, right? And just for you to understand how important is the gene ontology in general for research and all the annotations that we have there for research, if you just go to Google Scholar and quickly look for gene ontology for 2021, you will find that at least 2,500 papers have been written pointing to Google Scholar. And actually, these are only cancer papers using the gene ontology only this year. So it's a very important resource, and that's why we want to actually keep improving it. So now what we have here, this is actually how kind of like the data looks like. What we have here is a phylogenetic tree. This is a real phylogenetic tree. I won't pronounce the name of this family. Okay, that's the code from the Panther database. We have here, this is a Go function, Go term, which is associated with antiviral activity, okay? And the lines that we see here is actually the actual annotations on the genes, okay? So the genes, the modern genes that we actually observe are the tips on this tree, whereas everything that is inside of the tree to the left-hand side will be the ancestral genes, the parent genes, right? Which we don't know, but we infer that they are there, right? Other features that I can tell you about this tree. So here we have the leaves, right? These are the modern genes. Interior nodes are the parent genes. Each kind of point here that you observe is what I will call as an evolutionary event. And evolutionary events, we in general, we have two types. We have a duplication event, which are marked with a black dot here, or other, which could be, for example, an speciation event. The key of difference is that in duplication event, that means that essentially at that point in evolution, a new copy of a gene was generated. We had an extra copy of a gene. And what happens with that is what determines whether we observe a new function, we lose a function, or something else happens, okay? That's the tree. And regarding the annotations, as I was saying here, the blue line indicates, for example, that this gene here has this function that is involved in antiviral activity, whereas the red line here bar indicates that we have evidence to say that this gene is not involved in antiviral activity. So it's a negative annotation. And with this positive, negative, and negative annotations, and with the tree, we can make inferences using a model, a parsimonious model that I won't go into much details into that, but the predictions look something like this, okay? So again, we have the tree, the phylogenetic tree, we have the annotations, and here are the predictions that we made. Each bar represents the credible interval that was generated after feeding the model, and it's telling you essentially how well we did making our predictions. As you can see, in some cases we actually were pretty good when it comes to like accuracy. Here we have we had a negative annotations, and we predicted the negative annotations. And here, for example, we have positive annotation, and we predicted a positive annotation, saying that effectively this gene has a function. But what's more,
sorry for interrupting, but are you using a pointer, because we're not seeing it?
Oh,
it sounds like you are, but we're not seeing anything.
Yeah, let me see. Oops, I thought I was using a pointer. Sorry.
You may be using it, but we're not seeing it. At least
okay.
I'm not seeing it.
I think I can draw now. Let me try to see. Yeah, I can draw now.
There you go. Yeah.
Okay, sorry.
No worries.
Thank you for noticing that. Okay, so I was pointing at this point here, right? We have a negative annotation, and we made a negative prediction as well. We have here, for example, a positive annotation for this gene here, right? And we predicted that we have a positive annotation here. But what's more interesting from here is the places in which we have blanks, right? What we are saying here that at this point, even though we don't have any experimental annotation, our model is telling us that there's a high probability, a high chance that these genes here in this group here actually do have the function that we are trying to annotate, okay? And since this model is essentially bringing up like a borrowing strength from the tree, from the other annotations, in places in which we don't have that much information. So for example, like places like here, we don't do that well. So for example, here we're just saying that essentially it's a coin toss. And here, because of the structure of the model, because of there's like a duplication event, it's saying that it's likely to have a function, but either way, it's not as certain as in these other sets of annotations. So in this case, the AUC, which by the way, we tend not to use much because it could be misleading for this kind of problem, is 0.91. This is the Lip-Wanao cross-validation AUC, and the mean absolute error is 0.34, which is pretty good for this model. And all of these that I'm just showing you here is already implemented in the prediction, simulation, model fitting in our package aphylo. Okay. So aphylo, which is the name of the package and the name of the model. And by the way, this is the hex sticker. I think I'm using, this is the hex sticker, right? Which is what people is using a lot of these days with our packages. Brings several things to the table. So the first thing is it works on large scale. I will go into more details later on, but the thing is that usually when you see a literature dealing with phylogenetic modeling of gene functions, they usually tend to look at one tree, one family at a time. Instead of that, with aphylo, we actually were able to pull trees and pull and fit in pool data models in which we, instead of having one tree and a handful of annotations, we could have hundreds of trees with thousands or hundreds of annotations. In our paper, actually, we fitted a model within 5 minutes in which we included 131 different families with 1,300 annotations. The other nice feature that our model brings compared to others to the table is that its results are, we can interpret the results in a better way. So in other words, from what we have seen in literature, it tends to be a lot of these other models tend to essentially be kind of like what's the word? Essentially, they just try to, it's an algorithm essentially. Instead of having like a full modeling to do inference, they have an algorithm in which they go and just fix the parameters to values that they believe are the more credible and the ones that make more sense, and go ahead and make the predictions. In our case, when we fitted our model, our results actually were pretty good in the sense that besides of getting good prediction quality, with the parameter estimates that we obtained were very much aligned with what we were expecting from based on theoretical results. In particular, the most important here is that as far as we understand, the key evolutionary event are gene duplication events, and in the sense that whenever a function is lost or gained, it should happen usually in a gene duplication event, and that's what our model told us. Also, as I mentioned, this is significantly fast. So when we were writing our paper, we had to actually make some comparisons with other models, and during a benchmark, in order for us to fit the entire, make the predictions actually only for the entire set of 1,300 genes. The other phylogenetic state-of-the-art model that we were comparing against, it took us about 2 hours just to make the predictions, without mentioning how much time it took us actually to install and set up the entire thing, which was very complicated, by the way. Whereas using our model, aphydo, to make the posterior, calculate the posterior probabilities, it took about 1 second. So 1 second versus 2 hours, you can see there's a lot of difference there. And finally, accuracy. To my surprise, our model outperforms state-of-the-art phylogenetic models using with an improved AUC and also mean absolute error. So we bottom line, this new model aphydo, it's bringing a lot to the table. It's great, it's fast, efficient, and it's easy to use. But nevertheless, there are some things that we're not happy with, and because of that, we started developing another modeling framework that will overcome some of the main issues and assumptions that we are doing with the aphydo model. But first, let me tell a little bit about theory. Okay, this is from a paper, a review paper from Einan and Kogarshop in 2010 Nature Review Genetics, in which they essentially group up all the theories or most of the theories at the time that will talk about gene functional evolution. And here I'm just showing you one slide in which we are including one of the categories that they talk about, which they call as neutral fixation. In this case, this talks about when you have a set of genes. I think I'm still writing. Okay, yes, I am. You have a gene, right, in the population, and at some point it becomes to duplicate. So you start getting extra copies of a certain gene. After some point and neutral variation, neutral fixation, this new copy gets fixated in the population, right? So now each organism has this extra new copy. So what happens next is that let's say the biology has to tell essentially what is going to happen with this new extra copy. So are we keeping this new extra copy? Are we, I don't know, selling it, or are we dropping it? What are we going to do with it? And the fate that this extra copy faces, it could be various. It could be very different. So, for example, one that's called the neo-functionalization theory says that essentially once you get this extra copy, after accumulating enough mutation, it actually starts doing a different function. So you have the red function, and now this gene does this new copy does the green function. So you have a new function. The other thing that could happen, for example, is that which is called duplication, degeneration, complementation, which means that you got this extra copy of the gene, but then for ABC, like the potency, the quality of what the gene was doing is not good enough. So at the end of the day, these two genes do the same function, but they actually complement each other in order to get to the levels of I don't know, some protein secretion or whatnot, like that should happen. But then you have a third, another thing that could happen, which is called specialization or subfunctionalization, which means that imagine that this gene in particular that was duplicated actually had multiple functions. But now, since we have 2 copies of it, what happens next is that instead of like doing both functions, the gene actually now one gene does one of the functions and the other one does the other functions. So they become specialized in what they do. So that way you get to preserve both genes, the original and the copy. And like this, there are many other examples. One that I like is one that's called like in which you generate a new copy, but this copy becomes a pseudogene, which you preserve just to to protect. That's a theory, to protect the organism from like mutations. So that way the original gene gets to like protect the original function. And because and all of these that I'm showing you right now, this is very cool, but our original model could not actually include this, and I explain you why. So this is like a general overview of how can we see phylogenetic modeling or functional evolution. Here we are usually we are going to be talking about a set of functions. For example, we have function set of functions A and B, right? Which and the parent gene, and then you have its offsprings, the siblings. And for example, in this case, sibling 1 actually gained a function A, while sibling 2 preserved the state of its parent gene, right? What we were doing in our first model in aphylo was essentially a very simple way to approach this problem, in which we assume that both siblings and all the functions are conditionally independent. Meaning that instead of like looking at the system as a whole, we're just looking at pairs. We're just looking, okay, does the offspring gain function A from its parent to its offspring? Does he loses it? Does he preserve the function, so on and so forth? But always looking at one function, one sibling at a time. And that's unrealistic because, as I just explained you, it could well be that, for example, function A was gained after like as a result of a duplication of other gene, and as a result of function B, for example. Another way to model this, which is what most others do, is look at sets of functions instead. So in that way, instead of assuming that the functions are conditional independence, you only assume that the siblings are conditional independence, but you model the functions jointly. So it could be that 2 functions are associated, like correlated, or they move together or not throughout evolution, but you do that. But again, this is not like entirely true, right? Because what usually happens at duplication event, it's this phenomena that I have here, that one of the siblings would preserve the functional state of its parent. And this model here is not capturing that. So then, like the best way to do this, in which you could essentially capture all of this nonsense and complexities, is just modeling the whole thing, the whole process together without assuming any conditional independence. This is great. This is what we could try to do that, but this has a problem. If we wanted to fit, for example, a model in which we have 3 functions, we will need to then estimate the transition, the full Markov transition matrix, which for 3 functions only has 512 parameters. And that for some folks may not be that much, but actually trying to fit these models is not straightforward. So finding these many parameters is not easy. And even more, if we actually were able to find them, get some estimates, the interpretation is pretty awkward, right? So what can I say about, I don't know, parameter 66 when you transition from this to this? I don't know. So it's not out of the box. It's very complicated. So looking at this, I actually when I start to see the functional states, the representation of the data like this, it actually made me think about social networks, and this is why. So let me do like a quick turn. Switch of talk. So now I'm going to be talking about social networks. What we have here is that in social networks, when we want to analyze a graph like this, the lingua franca of social networks, it's called exponential random graph models. This is like the way that scientists, social scientists use to statistically analyze social networks. The key thing of these models is that instead of trying to make a prediction of each one of the ties that we observe here, right, we focus instead of on sufficient statistics. And what I mean with that is that when scientists are fitting these models, instead of trying to think, I don't know, how likely is A to connect to B, we think about how likely are, for example, is the friend of my friend to become my friend, right? So we are testing, looking at motifs. And some motifs in social networks could be, for example, also when you're looking at what's called homophily. So if we try to hypothesize whether individuals who have a shared interest, for example, are more likely to connect. For that, we are not predicting the actual type, but rather we're modeling the graph as a whole and reducing it to sufficient statistics. Social networks can be represented as a GEESE metric, which is what we have here, right? So each entry represents one of the ties. In this case, the white entries mean no tie, and the colored ones means that there's a tie. So for example, this here means that B is connected to A. So B is sending a tie to A. Here means, this means that B is connected to C. So B is sending a tie to C, so on and so forth. But the thing is that, as if you recall from the previous slide, when I looked at this, I could not stop thinking about, our evolutionary events. So just like this, we can actually represent evolutionary events as binary arrays, in which, for example, the first column represents the functional state of the parent, and the following columns represent the functional state of each one of its offspring. And with this, since we have this, I feel, okay, actually we can go and take all of the machinery that has been developed for exponential random graph models, which is a lot actually, and use this to model the transition events in gene functional evolution. So in other words, going back to this previous slide in which I have the model here, if we want to, instead of like estimating the full transition matrix in which we have 512 parameters to find, we could well go and reduce the whole evolutionary processes to sufficient statistic and fit a model in which you have, I don't know, 11 parameters, for example. So you have the function, the number of functional gains, number of losses, whether there's a neo-functionalization event, a sub-functionalization event, and whatever your imagination leads you to. And the great thing of this is that this is significantly easier to interpret. Yeah, so to fit from 512 to 11 parameters, it's a big difference, right? But at the same time, this is easier to interpret, right? Because if we know that neo-functionalization is, I don't know, positive about 0, it means that, okay, there's a higher rate of observing neo-functionalization or a lower rate, I don't know. So, but the thing is that you can actually make sense of the parameter estimates that you obtain from this model. Okay. Here are some examples of some statistics that we could include. So for example, if you wanted to include gain of function, that's just simply, I don't know, a row sum in the GEESE matrix. A loss of function, a row sum, 1 minus the row sum of the GEESE matrix. Sub-functionalization, neo-functionalization, we can go and define whatever statistics you want to fit this kind of model. Right. So how can we actually compute this? The way that we do it is using Felsenstein's pruning algorithm, which is the same algorithm that everybody uses when it comes to phylogenetic trees, which is a recursive algorithm in which you start from the bottom, from the leaves, and go and start moving towards the root of the tree. So that's how you compute it. But the key thing here is that from a difference from other models is that instead of using, for example, full Markov transition matrix or some fixed parameters, the way that we were doing in our fixed model, we operationalize this using this transition probability, which is defined here as the ERGM from the ERGM model, exponential random graph model. So here the key elements of this transition probability is that you will have a vector of sufficient statistics, right? Again, the number of gains, the number of losses, whether there's a sub-functionalization event or not, you can, I don't know, include a dummy that indicates that only one of the siblings actually change in evolution and things like that. And then you have the vector of parameters, which is of the same length of the sufficient statistics. And finally, this is the vector of parameters, right? And finally, you have this thing here, which is the normalizing constant. This bit here is very important because it is the case that you do have like a limit, right? Because if you want to make of this a probability, you have to normalize it by the support, by all the possible combinations. And the way that this grows is actually pretty fast. So it grows something like it's actually 2 to the n. The number of elements that it contains here it grows 2 to the n. So if, for example, if you have, say, I don't know, an array in which you have something like, I want to say, I did have a few numbers for this. If you have an array in which you have, I don't know, in between functions and siblings, you have, I don't know, 20 cells of an array, then that means that you are looking at a support size of 1 million possible combinations. Nevertheless, in our case, as you will see, I already implemented this very computationally efficient, so that's not going to be a problem. And on the other hand, besides of the likelihood calculation of the likelihood, we also have to compute, right, the posterior probability, which is also a recursive function, but instead of starting from the bottom, from the leaves, you actually start from the top and then climb down until you get to the leaf nodes. And the nice thing of this function is that it's kind of like, it's very straightforward. At least I think of it, you can interpret it very easily. So for example, the probability of observing a gene with a given functional state, I don't know, having function A and another function B, given the data is a function of its parent, what's the state of its parent, times all the possible ways in which you can go from the parent to this state of the offspring. That's kind of the way that you can think about this function. And all of this, it's already implemented and more actually on GEESE, on the package that I mentioned. So this is GEESE, just like a Philo, we do have a logo for GEESE, hex sticker, and it's called Gene Functional Evolution using SufficiEncy. And by the way, this is actually developed under the hood. It has another C++ template library that I'm also developing that's called Barry, which is a motif accounting tool that essentially allows you to count sufficient statistics in binary arrays. And if you're interested about social networks, you should take a look at this because it's very, very efficient as well. Okay, so what are the main computational features of this GEESE? Okay. First of all, it allows us to constrain the support set. Since I'm computing all the support of this summation here, right? We have to iterate through all the possible states. We can actually reduce this by telling GEESE, okay, no, I don't want to see transitions in which, for example, you go from having no functions at all to having all functions, or vice versa, or from having all the functions to not having any function. We can constrain the support set, and then reduces the computational load of the entire algorithm. It also keeps does bookkeeping of the support across the tree, right? Because the transition probability that I just showed you happens every time at every interior node of the tree, and in a tree you can have, I don't know, hundreds of them sometimes. So computing these hundreds of times could be very computationally expensive. Nevertheless, since a lot of these times these transitions actually are very similar across the tree, you can actually group them together and just reduce all the calculations to only one. So for example, here is an example with networks, for example, right? So here we have 3 networks which could be different, I don't know, from different persons, but they have the same structure. So because of that, instead of like computing 3 times the support, we only do it once, right? And then the other 2 just point to the original support matrix, so on and so forth. And this helps keeping the memory footprint very low, and it's also computationally faster. And finally, another feature that it's great is the fact that in all of these algorithms, you don't need to climb through the entire tree necessarily. You just need to climb through the induced subtree which contains the information. In other words, you go to the tree and see which tips, which genes have information, and do the climbing only considering those trees. So instead of, I don't know, going through hundreds of genes, you just go through a handful. So this also makes the calculations faster. So now the next question is, okay, how big can we go? I already told you that this could have some computational issues, right? In the sense that it grows 2 to the n. So the question now is, how big can we go? Well, it will all depend on what's the largest event that you observe in the tree. And what I mean with the largest event, it means like something like this. So for example, here we have, this is a very simple phylogenetic tree, right? We have, this is a reconciled tree, meaning that it's fully, each one of its interior nodes has only 2 offsprings, fully, right? So this node has 1, 2, this also has 1, 2, so on and so forth. And if we're looking at 2 functions, the largest event in terms of like the size of the array that we have to model, it's only 2 by 3, right? Because we have the parent that has 2 possible states, and then its offspring who also have 2 states, so it's 64. So the support size for this will be 64. That's like the largest thing that we have to compute. But then, unfortunately, things are not that pretty all the time. So in some cases, you will see that you can find what's called a polytomy in a tree. And polytomy essentially means that instead of having a bifurcating tree, at some point you may have some events that instead of having 2 offspring, have multiple offspring. So it's multi-bifurcating tree. And because of that, since we're modeling the transition, you have to take that into account. Here we have, for example, a hard polytomy, meaning that in this case, biologists and curators figured that at this point, it did happen that from this gene, 5 others actually split it. So it could be like a speciation event. So you have 5 new species from an original species. And because of this, since we are looking at 2 functions, we have 5 siblings. The size of this array is 2 by 6, meaning that in this case, we have possibly 4,000 possible states for the support, which it could be a lot, but it's not that bad. And finally, this is another example just for you to like think of. Here is kind of like very similar to the first state. We have also a reconciled tree, but here we have 3 functions. So instead of have a 2 by 3, we have a 3 by 3 matrix. We have 512. So now the question is, how big can we actually go? Well, from my experience, actually, you can go pretty big. Let me tell you a thing that happened to me a few days ago. I've been working with this like binary arrays for a couple of years now, and usually like enumerating, for example, a million possible cases takes a fraction of a second sometimes, like I don't know, of a few seconds. So when I was dealing with this, I was doing some simulations, and I was looking at a tree that I thought had like, I don't know, a sample size of 600,000 or something like that. But it took me like 15 minutes to do the calculation. And I kind of freaked out. I started to go back to my C code and see whether I have a bug. I started to like profiling the code, and I don't know, I had a memory leak or something. But it turned out that I was not looking at 600,000 possible cases only. I actually was looking at a polytomy that had 7 offsprings, and in a tree that had 4 functions, meaning that the support size for that event was 4.3 billion unique combinations. And that's why it took me 15 minutes. Now, if you think about it, 15 minutes for 4.3 billion is not that bad, and you can actually make a lot with that. Okay? But that's not usually the case of things that you will be looking at. So usually I would say that if you have a tree in which you have, for example, you can up to 9 functions, for example, in a reconciled tree, you can do that kind of analysis. But I think so. I'm not a biologist. I guess that biologists will never try to model together like 10 functions, or maybe I'm wrong, but either way, if they wanted to, they might be able to do it. Okay. So just to illustrate this, I'm going to show you a brief simulation in which I simulate an entire tree using this integrate this complex model, which sees the evolutionary step as a whole. And following that, I'll try to fit the parameters, the model, to see if I actually can obtain something. This is kind of like a, what's the name of this? Yeah, I don't remember the concept, but I think it's just to make sure that things are running right. So here I have 100 genes on a simulated phylogenetic tree. I only have two functions, function 0, function 1. Function 0 will be gained with some probability at a duplication event, but then function 1 is gained as a neo-functionalization event from function 0, also at a duplication event. So now I'm actually saying, I'm putting like this constraint that was not able to be included in aphydo at the beginning, in the previous model, because I'm correlating these two functions, right? I'm also saying that there will be a higher chance of gains at the duplication event. And finally, I would say that there's a very low chance of the root node to actually start off with the functions from the very beginning. I simulated this and then fitted this model using robust adaptive metropolis with a logistic prior centered at 0 with scale 2. So it's informative in the sense that I'm kind of like shrinking the parameter space so that it doesn't blow off. But either way, it's centered at 0, so it's not giving me any clue about where should be the actual parameters when I fit this model. And by the way, I fitted this using the R package FMCMC, which I wrote. So here are some of the results. I'm just showing you the trace of the posterior distribution from the MCMC. In the case of the gain and duplication event, a parameter, which I set it up for the simulation to be 2.5, meaning that there's a high chance of that happening. The model fitting worked pretty good. I was actually able to recover it mostly. In the case of neo-functionalization, it is a bit erratic. It is a bit erratic here, as you can see it. I keep forgetting that you cannot see my mouse, sorry. But either way, if you take the average of this or the 50th quantile, you will see that it's above 0 and it's close to 1.5. In the case of changes at duplication event, I also was pretty close to the actual population parameter. And regarding the changes at speciation event, which is the final parameter, in which I actually gave it a low chance of observing any changes at speciation events, it actually was pretty good as well. Nevertheless, in the case of the root node probabilities, we did a pretty poor job. But that's something to expect because we only have one observation, right? We only have one root, and this is what usually models like this tend to see, that the root probability is kind of like all over the place. Nevertheless, you can see that if you take the average of this or look at the quantiles, you will see that this is negative. So at least it's trying to capture that. So now, just to double check, essentially, I repeated this experiment 5,000 times. To see what I will obtain if I was getting the right parameters or not. So again, 5,000 times, simulating more trees, then obtaining parameter estimates using MCMC with a robust adaptive metropolis kernel, using a logistic prior, and do that 5,000 times. So, and by the way, each tree took less than 1 minute for estimating this MCMC. And these are the results. So this is the distribution of the parameter estimates from 5,000 experiments. The red diamonds that you observe here are the true parameters, the population parameters, and the boxes are just the box plot of its distribution. And as you can see, it's doing what it's supposed to do. It's doing a very good job of recovering the parameter estimates. They are covered, and surprisingly, the root node probabilities, even though I give no prior information to this whatsoever, it went negative. So it's tending to be negative, which is pretty good. So what kind of questions can we answer with this model? Well, I already mentioned some of them, but I could just give you some others if you are interested. So first, for example, this actually could improve prediction accuracy, right? Because we have a more complex model that is capturing more realistic traits of the evolution. We can also make a statistical inference of the sorts of, for example, which function came first? Is it function A or function B? When was the functionalization event more likely to happen, if we know that happened, for example? where functions A and B gain at the same time, so on and so forth. And you can answer all of this, or at least try to answer all of these sorts of questions using this modeling framework, right? But, and again, to emphasize that this is a modeling framework, right? So because ultimately, what goes into the model, it will depend on what are the sufficient statistics that you pick, that you design actually. So that's another neat feature of this new modeling framework. So now let me just go to the last of it. I'm going to just show you a brief data application in which I'm comparing a Phylo with GEESE using real data, experimentally annotated phylogenies, which we use for the paper that was published in PLOS Computational Biology. Okay, so this is from the paper that we published this year. And what we did is that we just re-estimated the models using GEESE with some prior information and compare those to re-estimations of the same trees and data using aphydo instead, with informative priors that we obtained from the paper. The data that we were able to use was a set of 77 experimentally annotated trees. We came to this number by essentially restricting the data sets that we could fit to only those in which you will have a support set less than 500 million, which is it's a big number, but and surprisingly, it actually went pretty fast. We did have some big trees, some big because we again the thing to blame here are the polytomies, because sometimes you will see that some trees may have an event in which you have, I don't know, 50 offsprings, 50 new nodes. So that makes things harder. So that's why we had to put that threshold there. So it's 500 million. And both models use quote unquote informative priors, right? So we again, so for the aphydo, we use the priors that we obtained from the paper. From GEESE, I use priors that I came up with. I don't know, priors that made sense to me from what I've learned working with this kind of data. Both were fitting using adaptive metropolis from the FMC-MCR package. Okay, and just to give you a glimpse of how this works, as you know me, I'm very interested about coding. So this is how the state of the art for this model it's coming up right now. So I have this R package that's called GEESE that generates GEESE objects. And here, after I created a data set model to fit, which essentially is a data set that I fill with real data, then I created the model following these steps. You can add terms like this. So for example, here I'm adding a term that will count the overall number of changes from parent to offspring, add duplication events, and add speciation events, so one for each. Then I am adding another term that counts how many of the genes changes, right? So in a case in our fully reconciled tree, it will be 0, 1, or 2, right? Because it could be that none of the genes changes, none of the offspring changes, one of them or both of them. But since we do have polytomies, it could be like, I don't know, from 1 to, I don't know, 50 or whatever number of polytomies you can find. I also added terms for gains and loss, add duplication events and speciation, one for each, for 0 and 1, gains and loss. And finally, this is another thing which I mentioned earlier on, which is I constrain the support set in order to fit these models that might have a support set of up to 1 billion, I don't know, possible combinations. I restricted this to only include those transition events, evolutionary events in which you could observe either no change at all, so from parent to offspring, you see they preserve everything, or up to 4 changes. Okay.
Are you talking about a single offspring there, or the sum of changes in all of the offspring ever given?
The sum of changes of all the offsprings.
Okay.
For example, I don't know, overall, one function was gained, one function was lost, or it could be, I don't know, you observe 2 gains, right? Because it could be that both 2 siblings gain off the same function, right? So essentially all the differences that from the parent to the offsprings. Does that make sense?
Yeah, are you going to tell us how many functions you're considering here?
For this case, actually, it's a mix because I have 2 and 1. That's the original data. Either 1 function or 2 functions. because again, all of this, I did all of this for 77 different data sets. So meaning that combinations of annotations and phylogenetic trees. So in some cases, you will have one function, in some others, you will have two functions. That's why it's different. Okay. But bottom line, this is just for you to have an idea. The thing is that the entire process is super flexible. You can actually build your own statistics, which is great, and it's going to be computationally efficient. So here's the punchline figure. What we have here, I'm just showing you the performance as a function of the mean absolute error. Okay, and actually 1 minus the mean absolute error, meaning that, I don't know, a value of 1 means that the model make a perfect prediction. And a value of 0 means that it was a completely completely off. On the y-axis, you will see the values of the mean absolute error from the aphydo, so the original model that we have. Whereas the x-axis shows you the statistics for GEESE. And as you can see, all of this area is actually, all of these models mean that these outperform aphydo, which is great. And the thing is that what I'm surprised is the fact that I just picked this model that I just showed you, kind of like a little bit out of the blue, without having fitted any model previously, without knowing what could be what would make more sense or not. And I just fit this model to all of the trees, the same model, and I got these results either way. So this is great, because again, computationally, it's more complex, but at the same time, the timing that I took to fit this model is actually fairly quickly. And finally, this is a table that is from the first model, aphydo, which is the thing that I have not discussed at all. So here, each one of these is just a single family. So it's a single gene, right? A single phylogenetic tree with sets of functions. But what if you actually fit all of these together in a single model? That's something that we already did in aphydo, and we actually proved that when you pull the data, it turns out that everything it improves. So compared to fitting the model separately, if you do a full data model, you can actually get statistically significant improvements from the baseline model. And in our case, well, since this is already computationally efficient, and I show you that I can do this fast, I've talked about billions of possible support sites, we can actually fit this using a flock. I call this a flock, and a flock is just a group of GEESE. And what I'm just showing you here, this is a dramatization of how it would look like, is that even though you can have, I don't know, dozens of hundreds of trees, since a lot of them actually share support, it will be just a handful of them that are actually contributing to the computational complexity of this. So this is something that I'm already have implemented, but I'm still testing it. So that's why I cannot show you results today. But what I can tell you is that this actually works. I tried it out in the sense that I was able to fit a model and obtain something in, I don't know, in a couple of minutes with hundreds of trees. So the take-home message is that from the start, what I started to tell you is that we are in a race right now to uncover what genes do, and this is key for biomedical research. As you know, again, if you go to Google Scholar, you look for gene ontology, cancer 2021, and you will see like thousands of papers using those resources. So we want to improve them, and because of that, a lot of scientists are doing that in an automatic way. But the problem with these many ways that they are doing it is that a lot of them have very unrealistic assumptions, as I discussed. And GEESE, which uses efficient statistics, it could be a way to overcome this, build a more accurate model, but at the same time, giving it more biological reality. So for the next steps, I pretend to further study its properties. I need to keep doing simulations and look at real data, fit the flux, as I mentioned, and find up actual real applications for this modeling framework. Because again, I need to, this is something that I'm very keen to do, because I know that in literature, you will find examples in which the sets of functions are compared together. but I haven't found any in particular that I could use for this, and I'm looking forward for that. And that's it. Thank you very much.
Thank you very much, George. I don't think I've ever given you a talk where you don't have this picture of the red and black dots.
Yeah. my thing, I guess.
We have a black box has just appeared on the screen towards the right-hand side. Any questions from anyone?
No, I think
Perhaps
it's very cool.
Sorry, go ahead, Kim.
No, Duncan, mine wasn't the question.
I was just going to say, I just looked up the collective noun for geese. Gaggle.
Well, so I found gaggle if they're on land, and skein if they're in flight, yeah, or a flying V or a wedge. So yeah, you're gonna have to choose whether your geese are airborne or not. Gaggle would probably better.
No,
Paul has a question.
Oh yeah, sorry, he's got his hand up. Paul.
I know, I'm trying to be polite.
We're not used to that around here.
That's right. Yeah, so George, this is obviously really great work. The one thing I was curious to ask about, and this is one of the nice things about reducing the parameter space for these massive numbers of transitions when you start talking about multiple functions and you can really start looking at evolutionary scenarios. One of the advantages, of course, is being able to interpret the parameters, as you said. But do you have some thoughts on actually how one would go about interpreting parameters? So what do the parameters look like that come out of the analysis based on sufficient statistics, and how would you go about interpreting?
You can use log odds to do that. Actually, there's a way. I just came out with this yesterday. This is very new interpretation of this. So you can doing the math. If you do you can directly compute this, translate them as probabilities. So for example, it could be given you will have I don't have any figure that I can show you right now, but you may have, for example, you want to see what's the likelihood of transitioning from having function B, not having function B, to having function B. Yeah. And you can compute the difference in the change statistics, combine that with the parameter estimates, and essentially multiply them directly will give you the log odds of the of the model. And then if you use the logistic function, that you can obtain direct probabilities that will tell you what's the likelihood of transitioning from a state to another state, including considering all the parameters, all the the modeling framework that you have. So yeah, that's a way of there's a way of directly interpreting this using as probabilities.
Nice. And
but that's non-trivial when you've got 2 to the n states, right? So you're going to have to summarize them in some fashion.
That's the key point. So if you do the math, all of those 2 to the n states go away, and you just end up with a vector sufficient statistic and the vector parameters. I have the math.
It's really going to be about interpreting what the parameter for a given statistic is actually reflecting in biological reality, right?
Right.
Checking for other raised hands in case we've had an outbreak of politeness, but I'm not seeing anything.
I have a question.
Oh yeah, there we go.
No, this is fantastic work. I love it. It's really great. One question I have is the rationale for going to the sufficient statistics is that the transition matrix has huge number of parameters. But then you talked about constraints, natural constraints, that could also apply to the transition matrix, making it naturally sparse. And I wonder, if you do that, whether wouldn't it be more interpretable to work on the original transition matrix as opposed to on the space of sufficient statistics?
Right. You could still do that, but the thing is that if you think about it, the thing with the sufficient statistic is that you can. This is a generalized modeling framework, so from there you can actually fit an equivalent version of a full Markov transition matrix, even if you constrain it. So yeah, that's what some people do. They just constrain the transition matrix in order not to estimate all the possible combinations, but nevertheless, the interpretation, it could be still a bit awkward. And the thing with the sufficient statistic is that you can essentially combine across possible like transitions. So it's more general. That's the bottom line. I mean, you could use this to go back, quote unquote, to the full Markov matrix, or you can go ahead and reduce this using other sufficient statistics. Does that make sense?
Yeah, totally. Great, thank you.
Paul, your hand's still in the air. Is it just stuck? Or...
There we go. I have to do something proactively. It doesn't know when my question's been answered.
Yeah, you'd figure, yeah, it'll just expire after a while.
Who designed this software?
Not George.
That's right.
All right. I guess last call for questions. If not, we're probably done. Thanks very much, George.
Yeah.
It was entertaining as always. And,
yeah, it's a lovely talk. Thanks.